I'll do one too.
Great. So hi to everyone who just joined as well. My name is Nina, and Joycelyn will also be presenting, and we're from the McCain Lab. So today we'll be talking about microscopy and histology, and we'll go ahead and start with microscopy. We'll start with an overview of our lab, my introduction, an introduction of my research, and then the microscopy, and then I'll pass it over to Joycelyn. So with drugs, what typically happens is that they are tested in a high throughput manner. So human cells can be seeded into multi-well plates and tested. But another way is for those drugs to be tested in vivo. But what happens oftentimes when moving from in vitro to in vivo is that the differences between human physiology and animal physiology makes it such that the drug doesn't act the same. So one example is with rats versus humans. The heart for a rat is 8 millimeters, but for a human, it's about 12 centimeters, and the heart rate is entirely different. So that might mean that a drug might work for an animal, but it might not work for a human. So what our lab does is we make organs on chips, and we aim to recreate different organs on cover slips so that we can see their mechanobiology or other features of those cells using a microscope. Typically, our lab uses what's called PDMS to mimic the extracellular matrix. So on the left, you can see a decellularized heart. And so what's left after you take those cells off of the heart is the extracellular matrix. And it's a scaffold so that the cells can attach to the scaffold and grow in a specific 3D organization. But it's hard to recreate that if you're doing it in a lab and you don't have access to a bunch of hearts that you can decellularize. So we often use PDMS to replicate the extracellular matrix. PDMS is something that you've probably heard of. It's used oftentimes in contacts, and that's because it can bend and it's optically clear and it's gas permeable, which are all things that cells need access to as well when you're growing them in vitro. So a little background about me. I am from Los Angeles originally. I went to Marlboro School. I went to the University of Pittsburgh for undergrad, and I studied bioengineering with a concentration in medical devices, and I got a minor in industrial engineering. Then after graduating from Pitt, I started working at Baxter Healthcare in their rotational development program. And in that program, I had 3 different roles over 2 years. I was in risk-based lifecycle management, which is essentially quality. I was a manufacturing supervisor, and then I was a researcher in research and development. Then once I finished that program, I stayed with Baxter, but I moved to Northern California, and I was a quality engineer for 2 years. And then after that, I decided that I wanted to come back to school, and so now I'm with the Laboratory for Living Systems Engineering. Here are some of the products that I've worked on, and some of, sorry, it's loud outside, and some of the devices that I've also worked on. So I've worked on fluid pumps that are used to pump intravenous fluids into a patient. I've worked on a fetal cranial shunt, which is used to, it's not an approved surgery at this point, but it's an experimental surgery to apply a shunt to a baby's head so that you can relieve the cerebrospinal fluid and prevent hydrocephalus from happening. I've also worked on flow seal and co-seal, which are flowable hemostats. So if a surgeon is operating and they're actively bleeding, you can apply these flowable sealants to the area to stop the bleeding. And then in the bottom left is Nextrone. It's one of the intravenous drugs that I worked on. And then in the bottom in the middle is a dialyzer that I worked on. The purpose of that project was to convert a dialyzer into a water filter. So if you have any questions about those, feel free to reach out to me in future. And now I'll talk a bit about my research. So my research is about the development of physiological assays to characterize the mechanobiology of uterine smooth muscle cells. The uterus is made of 3 layers. There's the perimetrium on the outside, the myometrium in the middle, and the endometrium on the inside. The myometrium in the middle is what's responsible for contractions during labor, but sometimes the myometrium doesn't contract the way it's expected to contract. In 2017, 295,000 women died from complications during or after pregnancy or childbirth. And one of those complications is dystocia, which is insufficient progression of labor. And that's when there are weak contractions of that myometrium that I was talking about. And that leads to surgical intervention in the form of a C-section. Have you guys heard of C-sections before? Yeah. But a C-section leads to longer hospitalizations and a higher risk of maternal death. Then there's also preterm labor, and that's when there are contractions before full term, and full term is typically defined as 37 weeks. And this can lead to health complications for the newborn, like it's possible that their lungs aren't fully developed, and so we try to prevent that to improve the outcomes for the child. So the mechanobiology of the myometrium is difficult to study. Some theories right now for the causes of reduced contractility include increased matrix rigidity and perhaps the combined effects of estrogen and progesterone. But to be able to study this in vitro, we have to use immortalized smooth muscle cell lines because it's hard to access tissue samples from a pregnant human uterus. Just imagine going around and asking a bunch of people for a sample of their uterus while they're pregnant. I don't think that that's super feasible. So that's why we used immortalized smooth muscle cell lines. And these cells were derived during pregnancy at 39 weeks. So they are relevant to the clinical conditions that we're talking about. So the aim that I'm working on currently is to see how matrix rigidity impacts the contractility of these human uterine smooth muscle cells. So it's difficult to measure the contractions of these smooth muscle cells directly, but the smooth muscle cell contractions are activated by levels of intracellular calcium. So instead of directly measuring the contractions, I can use flow4, which is a fluorophore, to indirectly measure the intracellular calcium, which indirectly measures the contraction measurement, or measures the contraction of those cells. So in the bottom left, you guys can see a time course study of intracellular calcium in PHM cells. So from the left, that's the beginning of the time course. And then at 50 seconds, oxytocin, which is a contractile agonist, is added. And you can see that the intracellular calcium increase, and then it decreases back down to a steady state value. So I wanted to recreate that study in our lab. The video that I'm about to show you was obtained in our lab using our fluorescent microscope. So the first section is A, and that corresponds to just obtaining a basal calcium concentration. Then now you can see that I added the oxytocin and there was a flash, and now it's going back down to a steady state value. And then I was able to use MATLAB on the right to convert the intensity values from that video into numerical values to be able to analyze those values. So based on that presentation, I wanted to know if there are any microscopy techniques that you guys picked out from any of the images. I'll scroll back and just give you guys time to brainstorm. I try to sing happy birthday twice, and that's how long I know to wait. But this one is bright field microscopy. And then I was able to use fluorescence microscopy here. So those are just two examples of microscopy techniques, and I'm just going to talk a little bit about how microscopy can help you gather data, how it can help you to quantify what you're looking at. So the general goal of microscopy is to increase the resolving power of human eyes and to magnify objects. And so the reason why I have that first part there is because it's not just to magnify objects, because imagine if you were to take a scan of something and then make it super large. That wouldn't be a microscopy technique, because it'd be super pixelated and you wouldn't have increased resolving power. So next I'll talk about light, resolution, and numerical aperture, and other commonly used methods of microscopy. So have you guys taken physics before? Can you use your reaction so I can just see and tailor the, I guess for the reactions, if you can use the, let's see, what kinds of reactions are there? Raise your hand if you've taken physics. Okay, those are thumbs up for physics. Joycelyn and Alex, you guys haven't taken physics? Just kidding. Yeah, so let's go ahead and start talking about light then. So light propagates as waves, but it's detected as particles, and this is known as the dual nature of light. And so I'm not going to go too much into detail for that, but if you're interested in learning more about the physics behind it, it's still something that physicists don't entirely agree upon, so it's pretty interesting to research. But just keep that in mind, that it can be considered as particles and as waves, and that's leveraged for the purposes of microscopy. So light as a wave, you guys have probably heard of the electromagnetic spectrum, and it ranges between 10 to the minus 14 to 10 to the 4 nanometers, but visible light is only within 400 to 700 nanometers. So what we see just walking around, it's within that small range. And the wavelength can impact the source of the light that is used for microscopy. And for light as a particle, we talked a little bit about resolution. The Rayleigh criterion is how people using microscopes are able to determine the resolution. So resolution is the ability to distinguish between two closely spaced objects. So if you have something that's resolved, you can see on the left that they appear as two separate entities. Then on the right-hand side, you can see something that's unresolved, so you can't necessarily distinguish them as two separate items. The Rayleigh criterion in the middle is that distance that defines where those two items can be resolved. Here's just another view of that. So on the last slide, I showed you guys like a 2D view. This is more of a top view of the ability to distinguish two closely spaced objects. So again, here you can see that those are unresolved and that these two are resolved. And so these individual points are often referred to as airy disks, and it's a representation of the point spread function. And so I'm throwing these terms out there, and if you haven't taken physics before and you're interested in this, I would highly recommend that you guys go ahead and Google that and learn a little bit more about it, because there are a lot of microscopy resources out there. But we'll go ahead and continue just based on this information. So resolution depends on wavelength and numerical aperture. The ability to identify two separate point spread functions, two individual points, again, depends on the wavelength and the numerical aperture. So let's go ahead and take a look at that. So here is, this website is really useful. It's the Zeiss website. And you can see here, they have these sliding scale bars that you can use and see how the wavelength and the numerical aperture impact the distance between these airy patterns. So if we make the wavelength really small, you can see that it's easier to resolve these 2 points. If we increase the wavelength, it might become more difficult to resolve those 2 points. And bringing that back down to the middle, if you have a low numerical aperture, it becomes much more difficult to resolve 2 points, but as you increase the numerical aperture, you can see the difference between the two. And again, if they're already separate on their own, then the wavelength and the numerical aperture will have less of an effect, and you'll still be able to resolve the two. But as the distance decreases between the 2 points, you want to be able to leverage the wavelength and the numerical aperture in your microscopy techniques. Let's go back. So here's just another view of what I just showed you guys about the difference between the wavelength and the numerical aperture and its effects on the resolution. So going back, the numerical aperture is the range of angles that a lens can accept. So it's just a convention that's used to be able to describe how useful the objective of your microscopy is. And you can change this n in that numerical aperture equation by changing the medium that you're using. So here you guys can see a bunch of different individual points, these point spread functions, and you see how it's fuzzy. And again, you can see that if you increase the numerical aperture, you also increase the resolving power. And a way to increase that numerical aperture is by changing your medium. So if you've heard of oil immersion microscopy, that's one way to increase your numerical aperture. Can I get a thumbs up just to see if you guys are there since I can't see you? Awesome. Okay, so let's go ahead and move on to some examples of microscopy techniques. So the way that the light interacts with your specimen is how you can define the different microscopy technique. Light that's absorbed, you can use bright field microscopy. If you're using phase contrast microscopy, the light is being scattered. And if you're using fluorescence microscopy, the light is being fluoresced. So starting with bright field microscopy, on the left you can see how the microscope is structured in order to accomplish bright field microscopy. Bright field microscopy is one of the more simple methods of microscopy. So if you've used a microscope, more than likely it was a bright field microscope. So you can see here that the light source is at the bottom. There's a condenser lens that condenses the light onto your specimen, and then the light goes back out onto an objective lens and then into the eyepiece lens where the observer can see it. It's really simple to use, but one disadvantage of it is that it has pretty low contrast. So you can see down in the bottom right that that's the image that I took with my PHM cells. So often what happens with bright field microscopy is that people use staining techniques to be able to increase their contrast and to be able to see their sample a bit better. Then for fluorescence microscopy, how familiar are you guys with secondary antibodies, primary antibodies, and fluorophores? Thumbs up if you have only heard about it just a little. Okay. Cool, so thank you. So fluorescence microscopy depends on fluorophores. And the way that I was able to stain my cells on the right-hand side was by using both primary antibodies and secondary antibodies. So the primary antibody is used first, and I guess that's why it's called a primary antibody. But so it goes in and it attaches to your antigen or whatever you specifically want to stain. And then once you have your primary antibody attached to what you're looking to stain, then you add a secondary antibody. The secondary antibody attaches to that primary antibody, and it's also a fluorophore, so it will light up depending on the light that is applied to it. So the two different types of light are called the emission light and the excitation light. So we can go ahead and look at those a bit more closely. So we talked about wavelengths of light before. So you can see here that light the excitation light can be within, or specifically, I used Alexaflor 488, which is phalloidin, and it comes off as green when it's in use. So the light that the microscope applies to my specimen is within this wavelength range, but then the light that is emitted is within a separate range of wavelengths. And different probe classes have different ranges for excitation and emission. So you can select your fluorophore based on what you want to image. If I wanted to image multiple things, I wouldn't want multiple fluorophores that emit within this range, because then I wouldn't be able to tell the difference. So here with my samples, I used 3 different dyes. I used DAPI, which dyes the nucleus, phalloidin, which which stains the actin, and which stains the F-actin, and then a different fluorophore that stains the alpha SMA. And those are just different components of my cells. And I wanted to show you guys these pictures because I don't want to say that science isn't art, because I feel like that's too cliche, but something that I'm working on right now is trying to figure out a primary antibody that'll work best with my cells, because there are numerous anti-alpha SMA fluorophores out there, but for some reason they're manufactured differently, so one might work better than the other. So if you can look at the image in the bottom right hand corner, the pink stains are pretty fuzzy, and what I wanted to be able to see was more fiber. So I know that the stain isn't working particularly well, or that my cells aren't working. So right now I'm assuming that my primary antibody isn't working quite well. So I'm shopping around for different antibodies. So that's just an example of a project or a troubleshooting technique that you might run into as a PhD student. And then the last type of microscopy I wanted to talk about is phase contrast microscopy. Phase contrast microscopy converts phase differences into amplitude differences so that you can see the topography of your cells. So something that's really great and useful is that you can combine these different microscopy techniques to give yourself some more information. So you can see that, can you guys see my mouse? Yeah, so you can see that for this one, you've got phase contrast, you also have fluorescence microscopy, and you can do an overlay to see both of those things and provide yourself with more information. Another cool thing about microscopy is that it can not just be used for these research techniques, but it can also be used for surgery techniques. So here's an example of fluorophores being used to image ovarian cancer, so that a surgeon can go in and identify cancerous cells. Here is another image that I learned about in our nanomedicine class during our fall semester. This was another imaging technique that was used to be able to see a tumor. So in this top row, you can see that it's kind of difficult to tell the difference between the tumor and the remainder of the tissue sample. But by using fluorescence imaging, it was easier to be able to identify the difference between the healthy cells and the tumorous cells. So that's another application of fluorescence microscopy. And I can send you guys more links in the sub-cohort Slack group if you guys are interested in learning more about how imaging techniques can be used for surgery. And with that, I will go ahead and pass along to Joycelyn.
Awesome, thanks, Nina. Let me see. Cool, so now I'm gonna pivot a little bit into histology. So does anyone have any questions for Nina before I start? Cool, it's pretty interlinked, so it should be pretty clear. So just also quickly, My name is Joycelyn. I am also in the Laboratory for Living Systems Engineering, and I'm just going to discuss my path as well. I grew up in Southern California. I went to high school in Boston and then came back to USC. I majored in biomedical engineering. I emphasized on mechanical engineering because I really liked seeing how different things work together. I was really attracted to like planes and cars and everything. So mechanical engineering was really the niche I was looking for. I also minored in art history just to balance out all my math and science classes. And I was lucky enough to study abroad twice due to a lot of help from my USC staff members. So I was able to do some research projects in Paris and Brisbane as well. Right after undergrad, I interned for a summer with Abbott as a quality engineering intern. And while it's labeled engineering, I didn't quite do engineering directly. So I was working within regulatory affairs, working with the Food and Drug Administration to make sure the Abbott products were meeting all of the federal guidelines as well as like international guidelines for other countries to get permission to sell them. So I learned a lot about like the commercial aspect of medical devices as well as how to distribute them to patients. After my internship, I continued at USC, so I was pursuing a progressive degree master's in mechanical engineering, as well as starting my PhD in biomedical engineering, which I was luckily able to do both at the same time. So within the lab, I'll talk a little bit about my research, but I wanted to highlight some of the research I did in undergrad as well, just to show you kind of why I was drawn to research and some of the different topics I was able to cover. So freshman and sophomore year, I was able to join the USC Biomechanics lab. That's actually not even in the engineering school because it's mostly biology and biomechanics based, but they use a lot of systems that engineers use as well. So I was able to join their wheelchair team as well as the golf swing dynamics team. They worked a lot with athletes to try to see the relationships between the human control system, which is the neuromuscular system, with the dynamic system of their skeletal muscles and bones, as well as how that interacts with the environment. So that was really awesome because I use a lot of sensors as well as force plates to see how the athletes pushed off in different directions for, shooting off to catch volleyballs or actually like their golf swing and how they rotated. So that was really interesting. That was a lot of sensor based and like cleaning up data. So I learned a lot there. When I was in Australia, I was able to work for a Gates Foundation funded project, which was incredible. They were trying to figure out how to deliver vaccines without using needles. There's a lot of issues right now with current vaccine delivery. If the vaccine is in a liquid form, it usually has to be refrigerated. So not a lot of countries have a robust, refrigeration system. So they were trying to figure out a way to make dry vaccines onto these patches that you could just deliver anywhere. So that was a really fun project to be a part of that really leveraged my mechanical engineering skills because I was tasked with finding a really good polymer to use as the base for these nano patches. So I was able to do material selection and work with a lot of plastics to see which ones formed the sharpest needles. And I was able to actually use histology on mice flank skin to see how far these little needles were able to go into the skin. So that was really fun. And when I came back for senior year, I worked in a implant retrieval lab where I was looking at spinal implants as well as knee and hip implants that were causing issues for patients. So if a patient needed a hip replacement and got a new hip, but it was really uncomfortable or if it was squeaky or it didn't really move in the right directions, the doctors would be able to retrieve it in another surgery and then send it to this lab to analyze what went wrong. So a lot of my time there was honestly cleaning the implants to make sure that they were clean, and then using microscopy to look at the surfaces of the implant to see if there was scratching and kind of like particles that were loose that would potentially damage the human tissue around it. So these were like the 3 projects I was able to get involved with undergrad. USC allows students to just reach out to faculty members. So I just kind of looked online to see what cool research was out there and contacted the faculty directly in order to score these positions. And as an undergrad, obviously, I was doing a lot of the grunt work stuff, but I think working within the lab and with PhD students, I was able to see exactly how research went and how labs were run. So it was very advantageous for me. I actually just recently completed my PhD as well. So I'm not going to dive into all the nitty gritty of it, but I wanted to also show how histology found its way into my research, kind of like how microscopy is important for Nina's research. So the title of my dissertation was Engineering Complementary in vitro and ex vivo cardiac microphysiological systems towards these larger goals of disease modeling and drug screening for humans. So I'm going to speak in the context of cardiovascular disease because that was mostly what my project was about. So just to give you some context, cardiovascular disease is the leading cause of death in adults in the United States and around the world. It passes cancer, respiratory disorders, and nervous system disorders. Cardiovascular disease also includes heart failure, which is caused by a variety of factors, either by heart attacks, where some portions of the heart tissue, as you can see in this purple gray area, where they die due to the lack of oxygen due to a blood clot in one of the vessels. And heart failure can also be caused by other acquired or inherited diseases. Because human hearts are extremely limited in their regenerative capacity, any kind of damage to the heart is often irreparable because the scar tissue that forms fails to maintain both the electrical connection as well as the mechanical connection needed for the heart to beat. Because of this, and because the heart can't beat as well, less blood is pumped to the rest of the body, and that ultimately leads to a decreased quality of life and ultimately death over a long time. So in the United States alone, this kind of failure affects over 5 million people and costs the US $116 billion annually. So it's really important for us to kind of address this and figure out ways to create better therapies. And you could also look at this graph. So despite this huge need for new drugs for heart failure and heart disease, the discovery of these new drugs has been steadily decreasing, as you can see in this like green line, while the investment into research and development has skyrocketed over time. So right now it takes approximately $2.56 billion to even get one drug into market. And even for the drugs that eventually do get approved, only 50 to 70% of the patients are cured. And often the other portion of it have unwanted side effects. So this kind of early stage drug discovery and safety assessment processes are really limited because of the current model systems that we are using today. So like Nina mentioned, these are our two gold standards, and there's a lot of parameters that we could assess these tools against. But most often the trade-off is ease of use versus environmental or structural complexity. So on the left, like Nina mentioned, we have these plastic petri dishes where we can grow single cell types or sometimes multiple cell types in a dish. They're really easy to use and very cheap. So that's why we could say that they're high throughput. We could have multiple conditions going at one time. However, the cells in these dishes are very different from human beings. So we as organisms, we physically move and our cells interact with chemical factors, electrical signals, and other environmental factors. So cell culture is often too simple to behave like humans do, and therefore they are limited in how they could predict the effects of drugs. On the other side, we have animals that we use, like mice and pigs, that are complete systems, and they could provide a whole organism response. So when they take the drug orally, we could see how it affects the liver, how it goes through the blood system and affects other organs. And while that's really beneficial, it's also limited because it's really difficult to raise these animals and keep them alive. So therefore, they are low throughput and very expensive. And as Nina mentioned, also animals, while they are great whole organisms, they're not exactly the same as humans. So yeah, mice hearts also beat much faster than human hearts. So to bridge these gaps between these two model systems, our lab and many labs out there are working on microphysiological systems, and they are kind of in between these two. they incorporate a lot of important features that you could find in native tissues. So in our instance, we want to incorporate a lot of functions that are native to what a heart really looks like. So they maximize the advantages of both ends of the spectrum, and they ultimately improve human relevancy, which is what we need for both disease modeling and drug screening. So looking at this, now let's connect it to biology, right? So for my project, I was able to design, build, and test 2 different types of devices. And just really quickly, the top row, it's a very simple platform where it has a glass cover slip. And for heart cells, we want to be able to electrically pace them, because that's exactly how our heart is able to beat in synchrony when it's pumping blood. So I was able to embed this transparent and clear electrode within the device so that we could actually stimulate them electrically with voltage and kind of track how these cells are contracting across the tissue. For the bottom one, it's separate. So I was actually studying zebrafish, and I was able to create a microfluidic device that was able to house 4 hearts at one time so that we could study longer term processes like regeneration. So in designing these tools, we have to look back at what we're trying to ultimately mimic. So I'm going to talk about the heart a little bit. I don't know if you have learned about the heart in detail in biology, but there are some like key features that we want to highlight. So this is the human heart, as you can see on the left. It continuously pumps blood throughout the body to deliver nutrients and oxygen to your tissues, as well as removing any kind of waste that your cells produce. It also has multiple layers, like the uterus. So it has the endocardium, epicardium, and the myocardium, which is the middle and thickest layer. And this thick layer is the one that really acts as the bulk of the muscle. And that's what contracts and pumps blood out. So each layer of this inside myocardium is filled up with cells like this. And these are elongated cardiac myocytes that are both electrically coupled. As you can see, they're like joined and mechanically coupled. And every time there is an electric pulse caused by certain pacemaker cells, all the cells kind of propagate this electrical signal. And when they like experience this electrical signal, they'll all contract at one time. So that's why your heart should always beat in a singular motion. If it doesn't beat in singular motion, it's called arrhythmias. And that's when different parts of the hearts are contracting at different times. So because all of these things are linked together, they're highly organized and they all contract in the same direction, which maximizes the force generation and then maximizes the output, which is the the blood being pumped out. So these kind of cues, so both the mechanical coupling and the electrical coupling, are key features that inform how we build our devices. And this is connected also to histology, because we are only able to see this organization because of histology, which is analyzing the structure of tissues. And just quickly, like why zebrafish and why I was interested in studying zebrafish. Human hearts, like I mentioned before, are very limited in how they regenerate. However, these adult zebrafish hearts can fully regenerate within 2 months if you cut off a piece of the apex, as you can see here. So we're actually looking towards zebrafish to see how studying their regeneration can be applied to human hearts. So with my device, I was trying to keep them alive over time. And this is actually where histology came in for my project. So all the images you can see on the left are different zebrafish hearts that have been cultured in different conditions. So some of them were cultured in my device, some of them were just cultured in petri dishes, and some of them were freshly explanted from the fish. And these are fixed, which means all the cells are no longer alive, but they are still stuck in the orientation that they were living in. Stained, so you can see different colors, and then sliced, so then you can actually see layer by layer what the heart looks like. And in this situation, obviously this is very qualitative. So when we're staining it, we could only score them, which is, instead of like measuring something, how long they are or how hot they are, we look at the qualitative properties of our stains, and then we're able to match them to scores just on our own by visually looking at it. So here you can see that I had different scores. So the score of 1 was no abnormalities. The score of 2, there's only abnormalities on the outside. And the score of 3 is that we actually see abnormalities going in. And using this kind of histological staining and scoring system, we're able to then determine, which condition was the best for keeping the structural preservation of the hearts. So you can see after 1 or 2 weeks of culture, the hearts in the device were more like freshly explanted hearts than the hearts kept traditionally in the dishes. So now I want to dive actually into histology and what histology looks like. So you can see this is the application that I use in my project, but histology is used all the time. So what exactly is histology? Histology is the study of the microanatomy of cells, tissues, and organs as mostly seen through a microscope. And it truly is a foundation of establishing how the function, the structure of these cells and how they're organized affects the function of them in our body. So definitely keep those 2 words in mind. And I think this is really important to also highlight. In biology, there's different hierarchies of organization. So you first start with cells, which many say is like the fundamental unit of an organism. So cells can be super specialized, but have also a lot of components within them, like a cell membrane, cytoplasm, organelles, and nucleus that are very important. The next layer are tissues, which is a unity of cells with a similar structure. As you see here is like a repeated muscle cell that as a whole have a very precise function. So this could be epithelial cells, connective tissue, muscle tissue, nervous tissue. Then the next level is a whole organ, and a whole organ, instead of a unity of cells, is then a unity of tissues, which have a more complex set of functions that define like the overarching function of that organ. So you can see the muscle tissue then becomes a heart. And then the next level after that is a system of organs. So a system of organs is united by similar functions that work together to perform critical body functions. So this is the circulatory system and how blood circulates throughout the whole body. And then lastly, it is ultimately like the whole organism itself. So the organism is a living thing that performs all of the 7 life processes. And I don't know if you all have learned this, like what defines what is alive and what's not alive, but there's 7 of them. So can anyone volunteer some ideas of how you would define if something is alive? I'll offer 2 of them so you kind of get what these qualities are. So again, we're kind of brainstorming what are the 7 critical life processes that define whether or not something is alive. So 2 of them would be sensitivity, like they actually have reactions to things, external environmental factors. And the other one is excretion. So they have waste products that are excreted out of them. Any other ideas of what would define if something is alive?
William says metabolism in the chat.
Yeah, metabolism. So that would kind of fall under like nutrition, like they have to have energy. So yeah, that's definitely one of them. How about something related to Nina's research?
Ethan says reproduction.
Yeah, so reproduction is also one. So that's already 4. The other ones are movement, respiration, and growth. So if things have all 7 of them, then that falls into our definition of what is alive. So again, I just wanted to highlight this hierarchical organization of biological principles and what organisms look like. So diving deeper into this, cells, again, is the smallest functional unit of the organism. And all the cells of the human body are eukaryotic, which means they are organized into 2 parts. So you can see a nucleus here, as well as a cytoplasm around it. So the cytoplasm is just like open space within a cell that contains more specialized subunits called organelles, which work like the little organs of the larger organism of the cell. So these organelles can contain these mitochondria, Golgi apparatus, ribosomes, nucleolus, all of these things that you'll also learn in the future if you haven't already. The nucleus in the center really functions like the brain. It contains the DNA blueprint of all the cells of your body and how they organize into tissues. All of the cells are then enveloped into the semi-permeable membrane, which means certain objects are able to pass through this barrier, but certain things like other fluids are not allowed through the barrier. And it kind of regulates what's external to it as well as internal to it. And it acts like a kind of like a border police, right? Like what can go in and out of the cell. They are organized into various different types and they have different functions. So you could have epithelial cells on the outside of your skin, fibroblasts, neutrophils that are part of your immune system, and many, many other types of cells. So these are the basic units. Then these cells self-organize into larger tissues. And there are actually 4 different types of tissues in your body. And they kind of form within a larger structure called the extracellular matrix, which, if you can imagine, is like the structure of the building, and all the people moving around it are the cells. So these tissues join together to form different arrangements in our body organs. So let's start with the nervous tissue first. These are made out of cells like neurons and glial cells that kind of propagate different signals. So it's involved with like movement as well as like reactions. There are little to no like protein fibers around it, and it's really pure. It's just like a link of neurons across your body. Neurons are classified into multipolar, bipolar, and unipolar. It's just the direction of where the signals go. And they exchange electrical signals throughout your body. Then the second type of tissue is epithelial tissues, and they cover external surfaces like the skin. So you can see that they're all aligned here to form a thick barrier. And they also align all of your inside organs, like the lining of your GI tract and everything also on the inside. So they are usually very, very dense because they actually form a physical barrier. And you can see that they're aligned like this. There's different types that they could form into. So sometimes they look like cubes, sometimes they're in tubes, but they're usually very, very packed in that particular fashion. The third type are muscle tissues. And like I was talking about with cardiac cells, they are categorized as skeletal, cardiac, or smooth. And based on their functional properties, they are either voluntary, like skeletal muscle, you voluntarily move your arm, so your muscles move, or involuntary, kind of like cardiac and smooth muscle. Your heart beats by itself, right? Like you're not actively telling it to beat. Despite their differences, though, they all have the same organization. So you can see that they're very much aligned into one direction versus these 2 other types of tissues. And they have things called actin and myosin in it to help it contract. And you could also see that there's sarcomeres in it too. The last one is called connective tissue, and these tissues separate and support the body functions. It consists of some cells, but it's usually mostly extracellular matrix. So that's like collagen, and it's very elastic. So it basically provides support and padding for organs and your joints. So depending on the type of the cells present and the extracellular matrix, the tissue can be either proper or specialized. So those are the 4 different types of tissues that you could find in your body. So then these tissues come together to form different systems in your body too. So these are just all of the different systems that work together to perform specific functions. So for the circulatory system, for example, they transport oxygen to the rest of your body as well as nutrients, and then they carry away waste, right? Then we also have the nervous system that controls things like sensation, your thoughts, your movements, and virtually a lot of the voluntary body movements. Then you have the respiratory system. It takes in oxygen and releases waste gas like carbon dioxide. You also have the digestive system that's all linked together. It breaks down food and it absorbs any nutrients that you may need, all the energy that powers your body. Then you have the skeletal system. It provides the structure and protects your internal organs. And then lastly, you have the muscular system, and it supports the body and actually allows it to move. So looking at all of these, histology comes in when we're actually trying to see it in more detail. So the tools for actually studying histology are becoming more diverse every day. And the most common tool for examining cells, tissues, and organs is optic. So like Nina mentioned earlier, everything to do with microscopy and actually using light to visualize it. So the first step in actually preparing the tissue is to get a fresh tissue. And then the second one is actually to slice it into very, very thin layers so that you could actually see it. So there are two main ways to do this. So if you take a fresh tissue, you could either freeze it with very, very cold temperatures so that it becomes a tissue block. And then you could use a cryostat to slice it into very, very thin sections. The other way is to fix it, and that's using a dehydration method to kind of remove all of the living portions of the cells and to put that piece of tissue inside wax and then also cut it into very thin sections so that light could pass through it. So you could do this with multiple different size tissues and organs, but you could see on the right, this is kind of what one slice will look like on a glass slide. And this is also stained, which I'm going to talk about now. So after preparing the tissue and slicing it, the tissue is stained for certain factors. Because tissues are normally colorless, we want to use different types of dyes to stain cells or portion of their cells under a microscope. These are all different types of stains that I just kind of pulled off a manufacturing website. So you can see that it stains for microorganisms, different types of connective tissue, all types of different things. The most common, though, is called H&E staining, and that's hematoxylin and eosin. So hematoxylin dyes cell nuclei blue, and eosin will dye the cytoplasma and the extracellular matrix pink. And that's what you could see in a lot of the previous images, if I could go back. So all of this is using H&E staining. They're all kind of blue and pink. So how histology is important is that it also intersects with pathology. So histology is just the study of tissues, right? But pathology is actually the study of disease. So histopathology is using histology to either identify disease or study how the disease presents. So a histology report describes the tissue that has been sent for examination after something realizes something is wrong. So if you've ever had a biopsy, that means a piece of your tissue was taken and then sent to a pathologist to then look at it. So I included two examples of what histopathology might look like. So the Gleason grading system is used to evaluate the prognosis of men with prostate cancer. So together with some other parameters, you could see the scale and what different tissue samples look like for the pathologist to then score. So then you're purely using this visual analysis to kind of tell people how aggressive they should be with their treatment of this prostate cancer. Similarly, you could look at tumor cells. So this is called a TNM classification. The T stands for tumor, the N stands for nodes, and M is metastasis. And this is the most widely used cancer staging system for cancer reporting. So for the T, you can see here, it refers to the size and extent of the primary tumor, which is like the initial biggest tumor. The N refers to the number of local and distant lymph nodes that are affected by this cancer. So you can see a score of 0, none of them have the cancer, but the score of 2, you can see that both distant nodes and the closest nodes are impacted. And lastly, M refers to whether or not the cancer has metastasized, which means the cancer has spread from the primary tumor to other parts of the body. So you can see here in 0, you can't really see this pink tumor anywhere, so it's the least score. Then 1, you can see 1 has gone here. And X just means it's really hard to identify whether or not these are actually metastasized tumors from the primary tumor. So while this is most widely used, it's not really used in common language. So I kind of wanted to connect it to stages, right? So I think most people who know people with cancer or have heard about cancer knows that it presents in different stages. So stage 0 means that abnormal cells are present but have not yet spread. But while it is not cancer, it could potentially be cancer. So maybe some moles that you might have might be stage 0, right? It's abnormal, but it's not cancer yet, but it could be. Stages 1 to 3 mean that cancer is definitely present, and the higher the number, and the higher the number means the larger the cancer tumor, and the more it has spread to nearby tissues. So it will fall in the ranges of like 1 and 2. And stage 4 means that the cancer has spread to the distant parts of the body. So this is just like an example of how histology can impact your prognosis. And this is something that is used daily, right? Like people go in, send away samples of their tissue, and this is how doctors are able to decide what treatment options to provide. So that's mostly it. Nina kind of covered the microscopy portion of it, but they're usually used hand in hand in all types of research and medicine. So for more resources, please head there. There are some ethical considerations too. Honestly, pathology is probably one of the least biased parts of medicine, because usually they don't include what the patient looks like in the samples that pathologists get. But obviously access to resources and access to pathology is very different. So I also included a link to some ethical considerations in pathology if you're interested in that. So that's all I have for today. Let us know if you have any questions.
Thanks for joining. I'll go ahead and stop the recording. Bye.
Thank you, everyone.
Thank you. Do we have a Zoom? Is
Yeah.
that a different link?
Yeah, we're going to switch over.